On behalf of FACP Echo Pakistan, I welcome all of you to this session on COVID vaccine. It's a very hot topic, and I'm sure I'm expecting more people to join in due course of time. I'm extremely grateful and really feel honored to have Adeel who is Professor of Medicine and Professor of Healthcare Policy and Research at Weill Cornell Medical College, and also he's the Vice Chair, Department of Medicine and Director of Clinical Epidemiology Research Unit at Hamad Medical Center in Qatar. He's also President and CEO of Innovations in Health Advocacy Research and Training, which is iHeart, in Washington, DC. हमारा इनसे बहुत गहरा ताल्लुक है, and I can proudly say कभी भी उन्होंने हमें ना नहीं की, and we keep bothering him again and again. और हमने we've learned a lot from Adeel in our echo sessions, and inshallah this journey will continue. Today he will be talking about COVID-19 vaccine, which is a hot topic these days, and he's also delivered this talk, I think, on a different forum a few weeks back, but we'd like to hear live from him, and then we'll have some burning questions for him to answer. So over to you, Dr. Adeel, please.
Thank you very much, Asad. Can you see my slides?
Yes, we can see your slides.
Gotcha. First of all, Assalamu alaikum, everyone who is attending, and I would like to thank Asad and Echo Pakistan for giving me this opportunity to talk to you today on a very hot topic, as Dr. Asad Chaudhry has pointed out. We'll be talking about SARS-CoV-2 vaccines and talk about some myths and realities. I have no conflict related to the current talk, and the question today is whether we should vaccinate or vacillate. In other words, should we wait for something better to come along, or do we have enough good candidates right now that we should go ahead and start vaccinating on a mass scale, and what are the potential ups and downs of vaccination? So that is the question. I'll be talking about 4 vaccines today in a little bit of detail. 3 of them have been approved through emergency use authorization. These are the Pfizer, Moderna, and AstraZeneca vaccine. And I will use Novavax as an example of another vaccine that is in advanced phase 3 clinical trials. And this will serve as a model for several others, like the CanSino and Sinopharm vaccines from China and the Sputnik vaccine from Russia. I may show a slide or 2 about the Chinese vaccine. However, not much data are available for those vaccines in published literature from phase 3 clinical trials. So unfortunately, I can't share much more on that. So let's start off with what do we have in terms of treatment for SARS-CoV-2 right now. So we have steroids, which have been shown to have some definite benefit in certain patients. Then we have remdesivir and IL-6, IL-1 blockers, which have been shown to have benefit in specific subpopulations. And perhaps monoclonal antibodies, although the data are not as clear on that. There are several agents which have absolutely no role in today's day and age in treatment of SARS-CoV-2 infection. These include hydroxychloroquine, azithromycin, lopinavir, ritonavir, interferon, convalescent plasma, and so on and so forth. There has been a lot of interest in ivermectin in the last couple of weeks or so. I'm not going to touch upon these things because today's talk is about vaccines. Now, the problem is that out of all these agents, the 4 or 5 agents in the center that I've shown you, only steroids have been shown to have a little bit of a mortality benefit. None of the other agents have shown any benefit in survival in patients with COVID-19 infection or SARS-CoV-2 infection. They have shown to decrease the severity of infection or reduce the viral shedding and so forth, but none have shown a mortality benefit other than steroids in certain populations. Hence the need for a vaccine for SARS-CoV-2. So this becomes very, very important that we need something that is effective, safe, at least in the short term, and which can be easily delivered to patients wherever it's needed. So that is what I'm going to talk about in the next 45 to 50 minutes or so. So I'll start off with a general overview of vaccine development and what types of vaccines are available for different diseases. And another hot topic in the last few weeks has been mutations and their effect on infection and vaccination. I'll talk about a few vaccines with the emergency use authorization or those who are at advanced stage of development. And then I'll end off with a series of commonly asked questions, because those are the things that keep getting asked, whether I'm in a social gathering or in a scientific meeting or giving a talk. Those are the things that I keep getting asked all the time. So I'm preempting some of those questions and we'll try to answer them. Let's start up with what are the types of vaccines that are available. So you can have about 6 or 7 different types of vaccines. You have DNA and RNA vaccines, and I want to be clear that no DNA vaccine has been approved for use in humans as of yet. RNA vaccines, 2 of them have been approved very recently, the last few weeks, and those are for SARS-CoV-2, and these are the Pfizer-BioNTech vaccine and the Moderna vaccine, and these are the ones we're going to talk about in a little more detail. You can have a vector-based vaccine, and many of them are in clinical trials for SARS-CoV-2, and then not many have been approved that we know of. You can have an inactivated vaccine, like a polio vaccine or a pertussis vaccine. You can have live attenuated virus like MMR or varicella or yellow fever vaccines, or the intranasal influenza vaccine. And you can have a subunit particle that is used as a vaccine like hepatitis B and human papilloma virus vaccines. Now, each one of them have advantages and disadvantages, but I'm not going to go into the details of that. I'll be happy to share this slide with anyone who would like to know. But why are vaccines important? Well, vaccines are very important because these are the only pharmacologic agents or agents that have been shown to eradicate disease. We can control and treat a whole bunch of other diseases in every system of the body, but the only diseases that have been eradicated from the world are infectious diseases for which we have had effective vaccines. And those include smallpox and polio, which is almost eradicated except 2 or 3 countries where it keeps popping up here and there. But with the vaccines that I've shown on this slide, we have had 99 or more % reduction in reported cases and a dramatic drop in mortality from all these diseases. So that is why vaccines are a critical tool for us to treat at a population level. Vaccine development, on the other hand, is a very tedious and a very lengthy process. It requires several years of early studies, clinical trials, animal studies, phase 1, 2, 3 trials, and then a very lengthy regulatory review by agencies like the FDA or the European Medicines Agency, and only then they go into production. And this process can take about 10 to 15 years. Now, SARS-CoV-2 vaccine development has been accelerated because of the emergency situation that we are in globally, and also because some of the preclinical work or the exploratory work had already been done, and we had those data on this platform for the last 5 to 10 years or so. We also saved a lot of time on regulatory processes. In other words, application process that would take 1 to 2 years was sped up to be completed in a couple of weeks. And those of you who may remember, actually President Trump's chief of staff called up the head of FDA and said that if you don't call the meeting and approve the vaccine on Friday evening and Saturday morning, you can submit your resignation. So regulators worked overtime, day and night, weekends, holidays to get these to us. This slide shows all the different types of vaccines. I've mentioned the vaccine types before, and almost all of them are being used in terms of clinical trials for SARS-CoV-2. This is the vaccine development landscape right now. We have at least 43 vaccines in phase 3 trials, at least 20 in phase 2, at least 20 another 20 in phase 3, and 3 of them have been approved under emergency use authorization as of yesterday. Now, there are many more in other early and preclinical development that I have not mentioned out here. These are the ones that have made to at least phase 1 and beyond. Here is a list of vaccines and their developers or the companies who are making them, who have reached at least the phase 3, which are human clinical trials on a large scale. And of course, leading are the Pfizer-BioNTech vaccine, which has received an approval in many countries, the Moderna vaccine and the Oxford AstraZeneca vaccines. Now, some other vaccines have had limited approvals in certain countries, but not from the FDA or the European Medicines Agency, which are the two most credible and widely respected agencies in the world. Since the first two vaccines to be approved were mRNA vaccines, I thought I would put in a slide to show what this actually means and how they work. So it's a very neat concept. So what you do is you take RNA from the virus, and it could be non-replicating or it could be self-amplifying, and I'll tell you the difference between the two. On the top here is the non-replicating one. The green one is the actual coding sequence, and you attach two a head and a tail to this, and then you put a 5' cap and a 3' cap. Now these ends, what they do is they make the RNA stable, because otherwise the mRNA is very, very unstable and would disintegrate within seconds or minutes. This stabilizes the RNA. You put them in a nanoparticle or a lipid nanoparticle, and these ends also help this construct to get into the cell and uncoat at the right time and be transcribed or translated later on while inside the cell. And once they express the protein that they code for, this protein is then extruded from the cells and elicits the immune response that is the hallmark of this vaccine. So this is how this RNA works. Please note that the RNA does not get into the nucleus. It gets transcribed in the cytoplasm in the ribosomes, and it does not integrate with the host cell DNA. Now, before we go into the vaccine itself, let's talk about the mutations a little bit. And some of you may remember that in early March, we had this big news about the D614G mutation that was found. It started one place and very quickly, within a month or two, became the dominant strain worldwide. More recently, we have had the B1.1.7 strain that I'm going to talk about in the next slide. But how often do mutations occur in SARS-CoV-2? Now, coronaviruses change more slowly than other RNA viruses because they have a proofreading enzyme that corrects fatal copying mistakes. Now, a typical SARS-CoV-2 virus accumulates only 2 single-letter mutations per month. This rate is about half that of influenza and about 1/4 that of HIV. And HIV, as you know, does not have a proofreading enzyme. Now, 2 SARS-CoV viruses collected from anywhere in the world differ by an average of just 10 nucleotides out of a total of nearly 30,000 nucleotides that we have. And however, despite the sluggish mutation rate, people have cataloged nearly 12,000 mutations in the SARS-CoV genome. So mutations are very, very, very common. The question is, how do they affect the virus and the host, and what do they mean? So D614G, which was the first big one, was discovered in February 2020 and became the dominant strain worldwide, was associated with higher viral load in the upper respiratory tract. However, it was not associated with increase in disease severity. The virus did replicate more efficiently and was more stable, though. Now, the B1.1.7, which is the big scare right now, is the result of 17 mutations in the virus. 14 are substitutions and 3 are deletions. Now, these mutations are in the ORF1AB. ORF is the open reading frame, and ORF8, but also the spike protein. Now, it is thought that they may have started in a patient who had long-term infection, which gave the virus time to adapt to sustain immune response in that patient. The patient may have received convalescent plasma as well. That further added to the survival pressure that the virus was under and mutated to survive that. However, the consequences of this remain largely unknown. We think that it may be more infectious. We also think that it is more replication competent. But again, it does not seem, at least so far, that it leads to more serious disease. We also do not think, at least at this point with these mutations, that the current vaccines will not work, and we'll talk about that. Another one that came up in the last few weeks is the South Africa strain, which is associated with 8 mutations in the spike protein. This one may also be more transmissible. However, again, the consequences of this mutation are still unknown and under study. This is just a list of the mutations that occur in B1.1.7 mutant, and I've listed all the mutations in the various domains. So let's move on to the first vaccine, which is the Pfizer-BioNTech vaccine. So based on phase 1 clinical trials, they found that this is highly effective in generating an immune response. So this moved on to phase 3 clinical trials very quickly. And the pivotal phase 3 trial that led to the approval of this vaccine is shown out here. They had about 18,500 evaluable patients who received 2 doses of vaccine and 2 doses of placebo. And these are the baseline characteristics, which we will not go into the detail, but they were relatively well balanced. Here's the bottom line, that only 8 people in the vaccine group developed symptomatic COVID-19 disease, while 162 people in the placebo group developed symptomatic COVID disease. Only 1 person in the vaccine group versus 9 person in the placebo group developed severe COVID. So this translates into a vaccine efficacy of 95%. So which is pretty remarkable, that a vaccine which was developed so quickly and so rapidly tested has such high level of efficacy in clinical trials. This efficacy was seen in the population overall that was vaccinated, but also in all the subgroups that they looked at. So age did not matter. So whether you were younger or older, the vaccine was still equally effective. Whether you were man or woman, it was still effective. In various racial or ethnic groups, it was still equally effective. And effectiveness or efficacy ranged from well above 90% on average. Now, the side effects are quite common. And I think this is something that we must tell our patients and the general public, that if you get these vaccines, you will get side effects. 80% or more of people got some local injection site pain, compared to only 15 to 20% of those who got the placebo. You're also likely to get a little bit of redness or swelling at the injection site. Fever and other systemic symptoms like fatigue, headaches, chills are also quite common. About half of the people will get at least one of these side effects. So we also need to reassure our patients who receive the vaccine that this actually may be a good sign that you are responding to the vaccine, that these reactions mean that you probably got your body mounted a robust immune response to the RNA that was injected into you. The Moderna vaccine is another amazing success story. And I just put in a timeline out here to show you how quickly these things developed. On January 20th, Moderna started working on a vaccine, and it rapidly went through clinical trials. Efficacy in monkeys were reported very early on. Phase 3 trials began within a few months of actually discovering the virus. And then in about 10 to 11 months or so, or about 11 months or so, from the date they started work on this vaccine, they actually had an approval from FDA under emergency use authorization. So an amazing success story that will be told for generations to come. Here's the flow sheet for the clinical trial, and this trial had about 14,000 evaluable patients in the vaccine group and about 14,000 in the placebo group. And here, 11 people in the first group, which is the vaccine group, and 185 in the placebo group developed symptomatic COVID disease. 30 people in the placebo group had severe COVID, but none in the vaccine group developed severe disease from COVID. So this translates to a vaccine efficacy of 94.1%. So almost exactly the same as what we saw for the Pfizer-BioNTech vaccine. So both of these are a highly, highly efficacious vaccine. This is just another way to show the same data, that after the 2 doses, the Kaplan-Meier curve departs sharply from the placebo versus the vaccine group. Again, side effects were very, very, very common, and most of these were local adverse events like injection site adverse events, but a fair number also got fever, headache, and fatigue. So most people should expect to get some of these symptoms. These are almost always self-limited and of short duration. Moving on to the Oxford AstraZeneca vaccine, which is the Shadox vaccine. So Shadox stands for chimpanzee adenovirus Oxford. They put all the things that went to make that vaccine into the name. And the trial number was AZD 1222. Now, this is not an mRNA vaccine. This is based on a replication deficient chimpanzee adenoviral vector. which contains the SARS-CoV surface glycoprotein and the spike protein gene. Now, this is a little more difficult to interpret trial because the study that they published, the key trial that published, was kind of an amalgamation of 4 different trials. And I've listed all the 4 trials out here and the characteristics of these. Together, these were about 5,000 to 6,000 patients in the vaccine group and about a similar number in the control group. And they also showed that the vaccine was quite effective when they looked at the overall efficacy of this vaccine. Now, the complication here was that they used several different dosing regimens and sequences. They used low dose, low dose, low dose, standard dose, standard dose, standard dose. Many permutations and combinations were used, which made it a little difficult to interpret the results. The overall efficacy was 70% or so, but when you look at the best group, which was surprisingly those who got a low dose first dose and a standard dose second dose of the vaccine, the efficacy was 90%. So the efficacy overall would be somewhere between 70 to 90% if they gave that dose. The side effects were again relatively common, but severe side effects were very, very uncommon, and none of them really were severe enough for hospitalization. So let's talk about the Novavax vaccine, which I'm using as a prototype for a bunch of other vaccines that are in development. Now, this is a recombinant spike protein in a nanoparticle. And it showed a high level of immunogenicity in earlier studies and in rodent and non-human primate models. This was a clinical trial design, and this is the antibody response that you see. So it's difficult to read through all this, but basically all of this shows is that you develop both a humoral response as well as cell-mediated response from this vaccine. Now, this is a phase 3 clinical trial, so we're hoping that some of these data will come out and will be published very soon. Now, the Chinese vaccines are in Pakistan and have been approved in certain Gulf countries. Example of that is CanSino adenovirus vector vaccine. Now, this vaccine is also a vector-based vaccine, but this is a single-dose vaccine, unlike all the other 3 that I mentioned before, which were 2-dose vaccines. Now, in early clinical trials, it also shows a high level of immunogenicity. So high level of antibodies developing, also developing a humoral, in addition to the humoral response, developing a cellular response. And side effects were mild to moderate, but again, very tolerable. None of them were really serious side effects. And the phase 3 trials for Chinese and the Russian vaccines have not been published, but there are press releases that we come across and proclamations from the government and the manufacturers, which say that the Chinese vaccines may have an efficacy somewhere between 75 to 85% or so, depending on who you listen to. And the Russian vaccine, the Sputnik, also has efficacy of some approaching 80% or so. However, I cannot verify these because these have not been published in peer-reviewed literature. So let's move on to the commonly asked questions, but let me show you a graphic that you will see, I'm sure you see a lot of information that you on social media, and it is very difficult to interpret or to determine the veracity or the truthfulness of that information. So all that goes in, and a lot of garbage comes out of that, garbage in, garbage out, so difficult to interpret. Our job is, as physicians, is to understand, read the actual clinical trials and the actual scientific information and make some sense out of that. And why is all that very important? To put that in the context, SARS-CoV-2 vaccines, as I've shown, have an efficacy of 95% or so. Compare that with influenza, which has an efficacy of 50 to 60 or 50 to 70% or so. Haemophilus influenza vaccine efficacy is about 84%. Hepatitis B, 80 to 100% depending on the age. Mumps, 88%. Tetanus and measles are much better. They have 97 to 100% efficacy. So SARS-CoV-2 vaccines are right up there among the best. I haven't shown some which have lesser efficacy as well, but for a vaccine that was developed so quickly and has gone through rigorous review and peer review and clinical trials, this is remarkable. So let's take on a few questions here now in the last 15, 20 minutes or so. So the first question that I keep getting asked is, which vaccine should I take if I have the choice, or if multiple vaccines are available? And my answer to that right now is whichever you can get at this point in time, you should really take that. The ones that are approved, of course. So, because at this point, especially if you are in the high risk group, meaning you are elderly, over the age of 60 or 70, or you have chronic medical conditions like hypertension, diabetes, significant obesity, any of the rheumatologic conditions, etc., you should really consider taking it right away, whichever is available for you. Next question, should I take 2 or 3 vaccines to increase efficacy? And my short answer to that is no. Incremental benefit would likely be minimal, if at all. You really can't get higher efficacy than 95% that you see with the Pfizer and Moderna vaccines. But side effects will definitely be additive. You get double the side effects, but no real benefit from it. Also, we do not know how this will affect your immune system or your immunity that would develop to that. Can I mix and match vaccines? This is a hot topic, especially in the last few weeks, where the United Kingdom came up with some strange recommendations, which they then retracted. That one thing they said was that you can wait for the second dose for a much longer period of time unless we've given everyone the first dose. They also said that you could mix and match vaccines. At this point, we do not recommend that. No authorities in the US, including the CDC, Dr. Fauci, NIH, FDA, none of them recommend that. And I think UK kind of retracted that a little bit as well. We do not know enough about this topic to recommend this, and do not recommend to combine the different types of vaccines at this point. If vaccinated, can I start traveling? Can I stop taking all the precautions like masks and physical distancing and hand washing and so forth? And the answer to that is not initially. And this upsets a lot of people, because they say that, what is the point of having a vaccine if I can't do all the things that I used to do? Remember, you have to wait till a large proportion of the population is immune for optimal control of the pandemic. Vaccines are not an immediate fix at all, but they are vital first, and early and a necessary step. So we request everyone to still follow the precautions till a large population is immunized or has developed immunity, and herd immunity levels have been achieved, so that the benefit can be passed on to the whole population and not just those few people. Another question that is very frequently asked is, I've already had COVID, should I still take the vaccine? And my answer to that is, if you have the vaccine available, yes, you should take it. And the reason for that is that the immune response after natural infection is variable, and it depends on the severity of symptoms as well. In other words, if you've had mild infection or asymptomatic infection, your antibody levels may be relatively low compared to those who have had a serious or severe infection. So it is unpredictable what your level of antibodies would be after natural infection. So we recommend vaccination, which produces a much more reliable and robust humoral and cellular immune response. So you have predictably high levels of neutralizing antibody from the vaccine. If I have allergies, can I still take it? And I think this stems from the fact that one person developed anaphylactic reaction at some point in time, and it sent chill waves throughout the world. So far, a few million people have received the vaccine, and we have not seen any true anaphylactic reaction from the vaccine that has been reported. Yes, you should avoid it if you've had allergic reaction to any ingredient of the vaccine, but this would be extremely rare. Also remember that there's a background rate of allergic reactions to any administered medicine or biologic. So please don't allow your patients or others or yourself to spread panic based on isolated cases. Now, we do recommend extra caution and monitoring in people who have had a history of anaphylaxis. So these people should be very carefully considered and monitored so that no panic spreads because of something like that happening. If I have immunosuppression, can I take the vaccine? There is no live or attenuated virus in the current vaccine. So it is generally safe to take this if you have any immune suppressive condition. In certain immune deficiencies, it is not known whether your body will mount a robust immune response, but there's no harm, there's no downside to taking it. You still might develop some level of antibodies. So generally, immune suppression is not a contraindication to the current vaccines that we have. Can children and pregnant women take the vaccine? Well, we don't have any data yet on both groups, but clinical trials in children have started. Currently, the vaccine is approved, the Pfizer vaccine is approved for children, for people down to 16 years of age, and trials have now started enrolling children between 12 and 18 years of age. There is not enough placental transfer of antibodies, which is another question that comes up in pregnant women. So the baby may not be protected from maternal antibodies, maternal vaccination, or maternal natural infection. How long will the protection last? And this is one of the difficult questions to answer. We do not know the answer to that because we haven't had enough of a follow-up after vaccination. But we do know that it probably lasts at least 3 to 4 months because that is the follow-up that we already have. And based on natural infection projection models, it is likely to last perhaps up to 1 year or maybe even a little longer. Antibody titers do seem to wane after natural infection after a few months, but after vaccination, they probably will last longer because you develop a more predictable and higher antibody levels with that. Another common question is, can I get COVID from the vaccine? And the categorical answer to that is no. With mRNA vaccines, there's no virus, live, dead, or attenuated. So there's no possibility of you getting COVID from the vaccine. Now, it is possible that the day you got the vaccine, you already had COVID, which was not manifest, or you were still in the incubation period, or you got it in the first few days after the vaccine when your antibody level had not developed. So that is a distinct possibility, although that would be very uncommon. However, remember, this is not getting infection from the vaccine. It is just because you had not developed your immune response. It does take a few days to a couple of weeks for immune response to develop in humans. Another question is, will I need every year or will I need booster doses after the first 2 doses or so? And this is kind of an extension of one of the previous questions, and we don't know the answer to that yet either. Immunity is likely to last at least several months, perhaps a year or so. And my hope is that by that time, everyone would be vaccinated, at least the first go, and there would be enough population-based immunity or herd immunity that the pandemic will be stopped. So we hopefully will not need boosters every year like influenza. But again, it's too early to tell right now. Can I still transmit COVID after being vaccinated? Short answer is yes, it is possible. Like I mentioned in one of the previous slides, that you may be in the window period where vaccine response has not occurred, you have not developed the antibodies, and you may harbor the virus. So it is possible to transmit. That is another reason why you must still follow precautions like wearing a mask, physical distancing, strict hand washing, till a large population has been vaccinated. What if I missed the date for the second shot or the second dose? You still have a fair window if you miss the second dose. So some people say that you have an extra 1 or 2 or perhaps more weeks after the first dose. You can still take it. And I advise people that if you missed it, take the second dose as soon as it is possible. If you miss, there's no need to start all over again. In other words, you don't need the first dose again. You just need to get the second dose whenever you can get hold of that. People ask me, isn't natural immunity safer than the vaccine? Shouldn't I rather just get natural infection rather than getting a vaccine? And answer to that is absolutely no. I mentioned earlier that antibody levels after natural infection are variable. They depend on the severity of initial illness, while vaccination provides reliable higher levels of protective antibodies. And also remember that with natural infection, there is a certain proportion of people who are going to develop critical illness and will die from it. And that number is not very small. The overall case fatality rate, if you just divide crudely the number of people who have died in the world in the last 1 year divided by the total number of infections that we have diagnosed, the crude fatality rate is about 2.5% to 3% or so. So in other words, 2 to 3 out of 100 people will die. And that, when you translate that to the entire population, that is too high of a number to be acceptable. With vaccination, there is no risk of disease, so you prevent all that critical illness. So no natural infection or natural immunity is not safer than the vaccine. People also ask me that if I'm young, healthy, and otherwise at low risk, why should I take a chance with the vaccine? I would rather take a chance of that I will not get an infection, or if I do, I will be at a low risk. Again, I would say that overall risk-benefit ratio heavily favors taking the vaccine. And I've mentioned several of these things in the previous slides. There is absolutely no guarantee that young, healthy, low-risk people will not get infected or will not get serious consequences. All of us, I think, have seen or heard of people in our families, in our friends, in our social circles who were young and otherwise healthy and who succumbed to severe COVID-19. On the other hand, serious consequences from the vaccines are extremely unlikely. Now, this is a big one. Will it alter my genes or my DNA? In other words, will I grow a tail, or will I grow horns on my head? And the answer to that is no, you will not. Because the mRNA vaccines, mRNA does not enter the nucleus, as I mentioned earlier. It does not integrate into the host genome. So there is no chance of this altering your DNA or your genes. Now, on the other hand, if you are looking for developing a tail or horns, that's a different story. You'll be disappointed. How will the recently reported mutations affect response to vaccination? I touched upon that briefly a little earlier on. These strains are likely more infectious, and they are more efficient at being transmitted from person to person. However, these currently reported mutations do not appear to affect the severity of illness. And they also do not appear to affect the immune response, at least for now. Now, if things change in the future, it's unclear right now. It's too early to tell. But with the current level and degree and the mutations that we've seen, this, while being more infectious, having higher viral load, more affinity to the ACE2 receptors, we haven't seen more serious consequences, thankfully. I will stop here and I will give it back to Asad. If there are any questions, I'll be happy to answer them.
Thank you.